Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A level Biology Unit 1 for January 2022. This is the Part 2 video. I'll put the link to the Part 1 video below the description box. Let us begin with question 5. Question 5 says, collagen is a component of the wall of the iodar. Describe the structure of collagen. So here I said, it is a fibrous protein. This is known from your textbook. It's a fibrous protein. It is composed of 3 polypeptide chains. These 3 polypeptide chains make up the triple helix, kind of something like that. They are polypeptide chains that are linked together by hydrogen bonds to make the triple helix. So they say held together by hydrogen bonds between the chains, and they have repeated sequences of amino acids with the highest content of glycine. So we have to know they are repeating sequences of amino acids, and glycine is going to be in high concentration. The next part says, explain the role of collagen in the walls of the iodar. We know that collagen is a highly structured material, so it's there to give strength. It gives the walls of the iodar tensile strength so that the iodar can withstand pressure of the blood as it leaves the heart. Remember, blood leaves the heart with very high pressure, so the iodar has to be able to withstand that pressure in order to deliver blood to where it's supposed to be taken at a high enough pressure. Let's continue. Here they say age and sex affect the type and density of collagen found in the wall of the iodar. They say these are thought to cause iodic stiffness. In one study, scientists investigated the effect of age and sex on iodic stiffness and the density of collagen in the wall of the iodar. The graphs show the results of this study. So here we can see iodic stiffness for the males as well as the females. All the male monkeys have more iodic stiffness in comparison to the young ones, and here we see again all the female monkeys have higher iodic stiffness in comparison to the young. When we come down here looking at the density of collagen, we can see young male monkeys have higher density of collagen as well as young female monkeys in comparison to their older counterparts. Another part, they say, in another study, scientists investigated the effect of age and sex on 2 types of collagen present in the walls of the iodar. The graphs show the relative proportion of each type of collagen in each group of monkeys. Here they're showing the relative proportion of type 1 collagen we see among male and female monkeys. So of course, for both, young monkeys have a higher relative proportion of collagen type 1 in comparison to their older counterparts, both in males and females. Moving on. Here we look at the results for the relative proportion of type 8 collagen. Here we see both male and female old monkeys have a higher relative proportion of collagen type 8 in comparison to the young counterparts. Continuing here, they say comment on the effect of age and sex on the types of collagen found in the walls of the iodar and iodic stiffness. So I will be referring to the different graphs one at a time. When we look at graph 1, I want to take you back to graph 1 and graph 2. In graph 1, we saw iodic stiffness. You can see the older ones have greater stiffness in comparison to the young. In part 2, we saw the density of collagen, and we saw the young ones had more in comparison to the older. So I say graph 1 shows that older male monkeys have more stiffness than young males, and also older female monkeys have more stiffness than younger females. The older males have more stiffness than the older females. I think you can see that. Going back here, we can see, yep, the older males have higher stiffness in comparison to the older females. Moving on. And then I said a small difference in stiffness between young males and females is also observed. Results in graph 1 are significant since error bars don't overlap. So if you look at graph 1 again, the error bars do not overlap, so we can say that the results can be believable. Now going to graph 2. Graph 2, remember, it's talking about the collagen. So I said males of all ages have slightly more collagen than females. You can observe that from graph 2. And collagen decreases slightly with age. Changes in stiffness appear not to be related to the density of collagen. Graph 3 shows higher type 1 in younger monkeys than in older ones, and there is more type 1 in females than in males at each age. Also, significance cannot be established or cannot be determined since there are no error bars. I want to take you back to that as well. Here, there are no error bars we can see, so we cannot see if results are significant or not. Moving on to graph 4, I said graph 4 shows more type 8 in older male monkeys, and there is a slight difference in type 8 between younger and older monkeys. Since we saw no error bars, so significance cannot be established. And overall, I say the type of collagen possessed appears to determine stiffness, since stiffness is associated with a decrease in type 1 and an increase in type A. This is my conclusion, combining the graph of stiffness and the last graph which showed the results from type 8 collagen. This brings us to the end of question 5. Let's continue to question 6. Question 6, the cell membrane controls which substances can move into the cell or out of the cell. Processes by which substances can move into a cell or out of a cell include diffusion, facilitated diffusion, active transport, and osmosis. A Venn diagram can be used to show the similarities and differences between diffusion, facilitated diffusion, and active transport. Statements about similarities can be written in the numbered parts of the circle that overlap. For example, statements about similarities shared by all three processes would be written in part 4, we can see here. So this is shared by diffusion, active transport, and of course, facilitated diffusion. So they're going to say, statements about differences can be written in the numbered parts of the circles that do not overlap. So here they say which part would contain the statement uses proteins. Proteins are used by both active transport and facilitated diffusion. However, diffusion, the substances will go directly through the phospholipids, so it is not going to use proteins. So the answer should be facilitated diffusion and active transport. Here we should have this part here, so the answer should be D. Moving on. Here they ask which part would contain the statement needs energy in the form of ATP. Of course, this is going to be active transport only, and if I take you back, it is going to be part V, which is part 5. So the answer should be whatever corresponds to that, and that is going to be the B. The next part says which part would contain the statement solids can only move down the concentration gradient. Moving down the concentration gradient only is going to be part of diffusion as well as facilitated diffusion, so it's going to be part 3. Remember, active transport, things can move against concentration gradient, so that is out. Let's continue. Here the answer should be a B. Part B says osmosis can be defined as the movement of free water molecules through a partially permeable membrane down a water potential gradient. Explain this definition. To be able to explain this definition, I have to look at the information that is given. Movement of free water molecules. Let me continue to molecules. And then they say through a partially permeable membrane and then down a water potential gradient. So down here I said movement of free water molecules because only free water molecules move. We have to know that water molecules that are attached to ions or other solids are not going to move because they will not be free. The next part says partially permeable membrane because it allows only movement of some types of molecules to pass, but not others, meaning it's going to be kind of selective. Then the last part says down a water potential gradient because water moves from a region of high water potential to a region of low water potential. That is how we define osmosis. Moving on. Here they say the photograph shows a Chinese mitten crab. They say mitten crabs spend most of their lives in fresh water and only return to the sea to breed. In an investigation, mitten crabs were kept in fresh water and then moved into the seawater. The water content and amino acid content in the muscle cells of these crabs were measured for 15 days after moving them from fresh water into seawater. The graph shows the results of this investigation. So let us see here. We can see the vertical axis has water content in the muscle cells. The horizontal axis has the time after moving mitten crabs into the seawater. From the graph, as more days pass, the amino acid content in the muscle cells increases, but the water decreases and then slowly increases after time. So let's go to the next part. Here they say seawater has a higher concentration of salt than fresh water. Explain the changes in water content and amino acid content in the muscle cells of the crabs in this investigation. Again, to refer you back to the graph, you remember we saw that initially the water content decreases and then slowly begins to increase, while the amino acid concentration increases and then kind of tries to flatten. So let's see what I wrote. I said, initially the water content decreases when crabs were first moved. Remember, they are moved in a solution where there is high amount of ions or salt. This is because the water passed out of the cells by osmosis. Then water content then increased from about day 4, and this is because the increase in the amino acid concentration in the cell. So as more amino acids increased in the cell, they became the solid, so water had to come in by osmosis. Amino acids increase, the increase was sharp at first, but then slowed down. This is because the insoluble proteins were broken down into amino acids by hydrolysis, and then the crabs eat more proteins, which are digested into amino acids, and these are taken up by the cells by active transport or facilitated diffusion from the blood into the cells. So water content in the cells increased because the soluble amino acids made the cytoplasm of the cells more concentrated than seawater, and therefore water moved in by osmosis. To take you back to the graph, we can see initially water goes down because remember, these crabs were put in a solution that had a higher salt content. So water moved and or left the cells by osmosis. However, as the amino acid concentration increased, remember they eat proteins. Proteins are broken down into amino acids that are taken up by the cells. So inside these cells, there was a higher concentration of solute, which is the amino acid, in comparison to the seawater outside or salt water outside. So then water began to move in by osmosis. So the water content increased as the amino acid concentration had increased. So this brings us to the end of question 6. Let us continue to question 7. Question 7. The risk of developing cardiovascular disease is affected by two groups of factors. Some are lifestyle factors that can be changed, and non-lifestyle factors that cannot be changed. Remember, lifestyle factors can be like smoking, inactivity, lack of exercise, and so on, while the non-lifestyle factors could be maybe the sex of the person. If you're female, sometimes females are more prone to certain diseases in comparison to male, or age. You cannot change the fact that you're older or you're younger, as well as genetics. You cannot change your DNA. So these are the non-lifestyle factors. These cannot be changed, while the lifestyle factors can be changed. So they're going to say methods to reduce the risk of developing cardiovascular disease include drug treatments and lifestyle changes. Which row of the table identifies one lifestyle factor and one non-lifestyle factor? So we know genetics, age, as well as gender. So that, and that are non-lifestyle factors. Remember, they are looking for which row of the table identifies one lifestyle factor and one non-lifestyle factor. So let's see here. This is going to be the answer because age is a non-lifestyle factor, while our body mass index is going to be a lifestyle factor. Moving on down here, they say explain why a person might have to take several types of drugs to reduce the risk of CVD. We know that cardiovascular disease is caused by a lot of factors. So I say because many factors cause CVD, different drugs treat different conditions, and therefore people have to take different drugs in order to be sure that they overcome every condition that could lead to those complications. Here they say nutritional studies have shown that dietary antioxidants can reduce the risk of CVD. Explain why antioxidants in the diet reduce the risk of CVD. Antioxidants reduce the presence of free radicals. So if these free radicals are reduced, there will be less damage to the endothelial lining of the blood vessels, and therefore there will be a reduction in plaque formation. Remember, these free radicals cause damage to the inner layer of the blood vessels. So if they are removed, it means there will be less damage, less atheroma, which could lead to plaque formation. So there will be less plaque formation due to the reduction in the damage that could occur onto the blood vessels. Here they say some studies do not assess the nutritional quality of the diet of the participants. Explain why the results of these studies have to be treated with caution. Here I said because diet has an impact on CVD. Remember, salt content. If somebody eats a lot of salt but that was not taken into consideration, they could have that risk factor, but it could be ignored, and they end up having cardiovascular disease due to intake of a lot of salt or as well as cholesterol. So here I said, high salt content causes high blood pressure and high fiber reduces cholesterol absorption. So the results will not be valid if these diet conditions were not put into consideration. Let's continue. Explain why changes in diet other than antioxidants can reduce the risk of CVD. Sometimes people have CVD due to the kind of food they eat. Not only is it antioxidants. So if you reduce the amount of cholesterol or fat intake, there is a chance that you could reduce the risk of having CVD. So I said, because diet affects many CVD risk factors, salt intake can lower blood pressure, lower fat intake can lower chances for formation of atheroma, and increased unsaturated fats can reduce cholesterol. So if somebody thinks they could have CVD and they take this into consideration, there is a chance that the risk could be reduced due to diet other than taking antioxidants. So this brings us to the end of question 7. Let us continue to question 8. Question 8. Sickle cell disease is caused by a gene mutation affecting the beta globin chain of hemoglobin. The mutation occurs in the 7th triple code of this gene. This mutation results in the amino acid GLU being replaced by the amino acid VAL. The table shows the sequence of bases in the first part of DNA in a person who does not have sickle cell disease. It also shows the corresponding sequence of amino acids in the beta globin chain. So here we can see, remember they told you in the 7th, so here they say 1st, 2nd, 3rd, 4th, and so on until the 9th. So here the question asks, give the 7th triplet code in the gene for beta globin chain in a person who has sickle cell disease. Remember the information was GLU is replaced by VAL, so it means we go to the 7th, this GLU is going to be replaced by VAL, and therefore the DNA is going to have GUG. So here I wrote GUG. Next they say, name the type of mutation that causes sickle cell disease. It is going to be a substitution mutation because one amino acid is taken out while the other replaces it. So that is a substitution mutation. Next they say the amino acid GLU is hydrophilic, meaning polar, and the amino acid VAL is hydrophobic, meaning nonpolar. Suggest why this mutation causes hemoglobin molecules to stick together. Remember they talked about polarity of GLU and the non-polarity of VAL. So I said the R groups of GLU and VAL may have different properties. VAL with hydrophobic R groups might form interactions with other hemoglobin molecules, while GLU may have repelled them. So part of the hemoglobin containing R groups turn away from the cytoplasm, and this could affect or could cause them to stick together. Moving on. Here they say in 2020 about 140 million babies were born in the world. About 305,800 babies are born with sickle cell disease each year. Estimate the ratio of babies born with a disease to babies not born with a disease. So if these babies are born with a disease, we have to subtract to find the number of babies who are not born with a disease. So here I said, babies born with sickle cells are those. Babies born without sickle cells are going to be that minus that, which gives us this one here. And then the ratio of the babies born with a disease to those not born with a disease is going to be that to that, which gives us 1, 2, 4, 5, 7. And that is going to be the answer here. The red blood cells in a person with sickle cell disease are sickle-shaped and less elastic. They also have a shorter lifespan than healthy red blood cells. These sickle-shaped red blood cells cannot carry as much oxygen as healthy red blood cells, and they get stuck in the capillaries. The graph shows oxygen dissociation curves for groups of people with sickle cell disease and those without the disease. The shaded areas represent the range of values for each group of people. So we can see here, vertically we have the percentage of hemoglobin saturated with oxygen, and we have the partial pressure of oxygen. Let's continue. A P50 value is the partial pressure of oxygen that results in 50% saturation of hemoglobin. They say calculate the largest difference in the P50 value between a person with sickle cell disease and a person without this disease. They want you to give your answer in appropriate numbers of decimal places. So let's go back to the graph and observe that. Remember, P50 is 50% saturation with oxygen. Since they want us to find the largest difference, I drew a straight line at 50% saturation, and then I got the greatest difference, which should be this value minus that value. Looking at my graph, this value here is at 4.4, and that value there is at 7.8. So the difference should be 7.8 minus 4.4. That should be the greatest difference. So let's go back here. You can see I said 7.8 minus 4.4, which gave me 3.4. And that is my answer here. The next question says, identify two conclusions other than the difference in P50 values that can be made from this graph. The first conclusion, let me take you back here. We can see that people without sickle cell always have a higher percentage saturation with oxygen at all points. You can see at this point, you can see those ones have higher. And when we go to this point, you can see there is going to be 25%, and this is going to be a little bit lower. So they always have a higher percentage saturation with oxygen at specific partial pressures. So that is the first conclusion I saw. The second conclusion is that there is a wider range of saturation for people with the disease than the people without the disease. And again, I can take you back here. These are the people with the disease, and you can see there is a greater range of saturation in comparison to people without the disease. So those are the two conclusions I had here. Moving on. Here they say the lifespan of red blood cells in a person with sickle cell is 11 days. This is 9.17% of the lifespan of a healthy red blood cell. calculate the lifespan of a healthy red blood cell and give your answer to the nearest day. So I said, let X be the lifespan of a healthy red blood cell. Remember, they told us 9.17% of a lifespan of a healthy cell is going to be 11 days. So I said, let that lifespan be X. So 9.17 divided by 100, which is that, times the lifespan of a healthy cell is going to be 11 days. Then I made X the subject, which should be 11 times 100 divided by 9.17. And I got 119.956. To the nearest day, it's going to be 120 days. That is my exact approximation. Part 4, they say sickle cell disease can result in death. Explain why the changes in the structure of hemoglobin and the shape of red blood cells can result in death in a person with sickle cell disease. Here I said, changes in the structure of hemoglobin cause hemoglobin to bind to less oxygen. Also, changes in the shape of red blood cells decrease the surface area for oxygen diffusion. Therefore, less oxygen will be available to the cells in the tissues for aerobic respiration. So less aerobic respiration can cause organ failure, and if this occurs in major organs like the heart and the brain, a heart attack or a stroke would result in death. This brings us to the end of question 8 as well as the end of this paper. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.